Hi class, this chapter is on chapter 21, which is on blood, and we'll first talk about the components of blood. We can divide whole blood. If we take a blood sample out of a patient, we can centrifuge it down. That means spin it down extremely quickly to reveal three components of blood. The erythrocytes make up about 44% of a blood sample. They're very dense, so they'll fall to the bottom of the sample vial. The buffy coat is the middle layer containing leukocytes and platelets. This forms about 1% of a blood sample. And the plasma is the straw-colored liquid that makes up about 55% of blood, and that will float to the top. The erythrocytes, the leukocytes, which are your white blood cells, and your platelets are all collectively referred to as the formed elements because they are the cellular components. So here is the whole blood separation and composition, taking a blood sample out of a patient, centrifuging it down, so spinning it extremely quickly, and then the blood sample will be separated into these three components. The plasma will float to the top. That makes up about 55%. Plasma is mostly water and some proteins and other solutes. Then the buffy coat is this middle kind of clearish colored fluid layer in between. This will make less than 1% of whole blood. And this will be made of your platelets, which help with blood clots, and all the different leukocytes, which are your white blood cells that help with disease and infection. And then the erythrocytes, the red blood cells, will fall to the bottom of the sample, and they make up about 44%. So your functions of blood are transportation. The big thing that blood transports are your oxygen, carbon dioxide, your hormones, your nutrients, other cellular wastes. Blood helps to regulate body temperature, pH levels, fluid levels in your cardiovascular system. And it's important to note that normal blood pH is around 7.4, with the range being a little higher and lower than that. Functions of blood also include protection with your leukocytes or your white blood cells. They will guard against infection by triggering an immune response to antigens, which are substances perceived as foreign to the body, and your blood will carry antibodies in the plasma. So here are the major functions of plasma and the composition of blood plasma. So your plasma carries nutrients, proteins, and hormones through the body to be delivered. It will also carry water, salt, and enzymes. And it also carries waste products derived from cellular metabolism, like CO2 and urea. And you can see kind of the plasma breakdown shown in this table. The formed elements consist of erythrocytes that transport respiratory gases, CO2 and O2. They make up the vast majority of the formed elements. And then leukocytes help mount an immune response. Platelets help with blood clotting. And you can see all of these formed elements with a blood smear underneath a microscope. And you can take a sample of your blood, kind of a fingerprint prick, place a drop of the blood on a slide, spread it out, place a stain on it, and then underneath a light microscope at about 640 times magnified, you can see the red blood cells make up the majority, the erythrocytes of the sample, and then you'll see a few leukocytes thrown throughout the sample as well, with the platelets being the extremely tiny specks. Here are more kind of the hematocrit is the percentage of the volume of all formed elements in the blood. Clinically, it's the percentage volume of erythrocytes only. So the hematocrit will be measured as a percentage. In the clinical setting, it's a measurement of all the red blood cells only, the percentage. It will vary with age. Children will be a little more variable, and also your gender, your sex. Males, hematocrit is slightly higher on average. And this is a great table just summarizing the characteristics of the formed elements, the diameter and size. So the erythrocytes will be kind of in the middle. Your leukocytes can be 1.5 to 3 times larger than an erythrocyte. And then the different functions of the three, their lifespan, so how long they are able to survive in the blood, and their density or the average number per microliter. Erythrocytes are commonly called red blood cells. They're not true cells because they lack a nucleus and organelles. They're about 7.5 micrometers in diameter. They have a biconcave disc shape, so they look like a donut, but without the middle. As they pass through extremely small vessels, they have to line up in a single file, and we call that term a rouleau. And this is a picture of a rouleau, the red blood cells lining up in a single file as they're passing through a small diameter, probably a capillary. Hemoglobin is the oxygen transport protein in the blood. So every erythrocyte contains about 280 million molecules of hemoglobin. Hemoglobin transports oxygen and CO2. Oxygenated blood means when the hemoglobin is maximally loaded with oxygen, and deoxygenated blood is when some of the oxygen is lost and carbon dioxide is gained. Each hemoglobin molecule consists of 4 globin polypeptides with 2 alpha and 2 beta chains. So this is just the protein structure of hemoglobin. This is showing kind of one of the chains with the iron heme group in the center. And each chain can hold 1 oxygen molecule. So there's 2 alpha globin chains and 2 beta globin chains, with them each being able to hold 1 oxygen in the middle. Each of the 4 globins possesses a heme group containing an iron ion, which will actually bind to the oxygen. So each hemoglobin molecule can then bind a total of 4 oxygen molecules. And we just said that each red blood cell has about 280 million hemoglobins. So that means that each red blood cell carries more than 1 trillion molecules of oxygen. The erythrocyte life cycle is about 120 days. So what is that, about 4 months? Old erythrocytes are phagocytized, so kind of broken down and degraded, pulled apart in the liver and spleen. The heme group is converted to biliverdin, which is then converted to bilirubin. And bilirubin will go on to become part of bile for your digestive system function. Bile is important for digesting your fats in your small intestine. Iron is then transported by transferrin to the liver and stored by ferritin, and erythrocyte proteins are broken down into free amino acids. So this just shows you kind of how erythrocytes and the components of erythrocytes are recycled after 120 days, their lifespan. They will be phagocytized in the liver and spleen. The heme components of the blood will be recycled. The heme will be converted into biliverdin, eventually into bilirubin and bile, and then the iron will be transported in the blood by the protein transferrin and stored by the protein ferritin in the liver. Leukocytes then are your white blood cells. They will possess a nucleus and other organelles and will be much larger than erythrocytes. They help initiate an immune response and defend the body against pathogens. Pathological conditions can cause abnormal numbers of leukocytes. Leukopenia means a reduced number of cells, and leukocytosis means an elevated number of cells. The leukocytes' flexibility and motility help them get to the pathogens. Diapedesis is the word we use for the process of them squeezing through a blood vessel wall to exit the bloodstream to get to a tissue. And chemotaxis is the process of moving towards chemicals that are found at the site of infection. You have 5 types of leukocytes that we divide into 2 groups. We call them granulocytes or agranulocytes. You have 3 types of granulocytes that we'll go over and 2 types of agranulocytes. So these are the 5 different types of leukocytes. Here are the 3 types of granulocytes. They have granules within them, kind of these darker staining specks. And then here are the agranulocytes. The monocytes are extremely large. Here are the lymphocytes, about the same size of a red blood cell. The neutrophils are the most numerous leukocyte in the blood. They have a multi-lobed nucleus. The eosinophil has a bilobed nucleus, and here's the basophil. So here's a good kind of characteristic table listing all of the leukocytes and their characteristics and their functions and their approximate kind of percentage in the blood. So the neutrophils are the most numerous, followed by your lymphocytes. And here you can read through the functions, a lot of them. Some will produce antibodies, some will release histamine, some will phagocytize pathogen or antigen antibody complexes. Some will be macrophages, which will phagocytize other pathogens. So here's the neutrophil. It's the most numerous white blood cell. Its function is to phagocytize and destroy bacteria. It's a nucleus with 2 to 5 lobes. It's with small granules and about twice the size of the red blood cell. Eosinophils compose of just less than 4% of all white blood cells. They play a role in ending allergic reactions, parasitic infections, and they have larger reddish granules about twice the size of a red blood cell. A basophil has large blue granules. It's about twice the size of a red blood cell. It's about 0.5% of all leukocytes. It functions in inflammation mediation, so it will release heparin and histamine. And then the agranulocytes, we have lymphocytes, which compose 20 to 45%. They're very important cells of the immune system. You can have T and B lymphocytes, which you'll learn about in physiology. They are incredibly important in fighting infectious organisms and producing antibodies, and they will act against a specific foreign molecule and antigen. The nucleus fills most of the cell, and they are similar size to a red blood cell. And here are monocytes. They kind of have this kidney-shaped nucleus sometimes. They're the largest leukocytes with no granules. They will transform into macrophages, which just have the ability to kind of eat up anything foreign. So they are phagocytic cells with large particles. Platelets then, we'll kind of end here with platelets. They are small, irregular membrane enclosed cell fragments. They do not have a nucleus. They're produced in red marrow from cells called megakaryocytes. So platelets are kind of just cell pieces that break off of these larger megakaryocyte cells. And platelets are very important in blood clotting, and thrombocytopenia is an abnormally small number of platelets. So here's a look at where platelets are formed in the red bone marrow. You can see here the megakaryocyte underneath the light microscope, and the megakaryocyte will kind of be connected to a blood vessel, and pieces will break off of the megakaryocyte, and those are the platelets. They're small, irregular, membrane-enclosed cell fragments. They do not have a nucleus. They're produced in red bone marrow from cells called megakaryocytes, and they are involved in blood clotting. And you can see them here. They're much smaller than the red blood cells and the leukocytes. Here's a look at a blood clot. So eventually platelets will attract these fibrin strings, and they will go ahead and kind of form a clump or a plug, which will trap erythrocytes or the blood to try to form a blood clot. Hemopoiesis is the production of formed elements. They are continually produced in the red marrow, and we call this hemopoiesis or hematopoiesis. They will all begin with a pluripotent stem cell called a hematocytoblast. And from that hematocytoblast, they will form either the myeloid line or the lymphoid line. The myeloid line will form all of your erythrocytes, megakaryocytes, and all the leukocytes except your lymphocytes. And the lymphoid line forms all the lymphocytes. And again, remember the lymphocytes are incredibly important in the immune response. And that's the end of this slideshow on blood. As always, come to my office hours if you have questions on Sunday nights at 8. And I hope you guys are all staying healthy and well.